Thank you very much, Krista. And I want to thank the organizers for inviting us to share our work. So I've been given 5 minutes to explain to you what my lab's major interests are. If I could have the slides. So as was briefly mentioned, the long-standing abiding interest of my lab has been G-protein coupled receptors. So these are the receptors that transduce the signal for most of the neurotransmitters, neuropeptides, and many other molecules. And our interest has been particularly in the brain. My group, along with my colleague Brian O'Dowd, we cloned over 70 of these human G-protein coupled receptors. But our long abiding interest has been in the function of dopamine. Dopamine functions in the brain and the G-protein receptors that mediate dopamine functions. And of interest to some of you, 4 of the 5 known dopamine receptors were cloned in the Department of Pharmacology here at the University of Toronto. So that's quite an achievement. So dopamine functions in brain are myriad, very important, from executive function involving learning and memory to reward and reinforcement, motivation, and arousal. And our particular interest is not only the physiology, but also the major links to neuropsychiatric disorders, namely that of depression, anxiety, drug addiction, and schizophrenia. So if you look at dopamine receptor localization in human brain, there are many regions of brain where dopamine receptors are present. Of particular interest to us with the neuropsychiatric connection is dopamine receptor expression in the limbic system and in the cortex and in the amygdala. These regions of brain are intimately tied to neuropsychiatric disorders, and therefore the functioning of these receptors in these regions have been of great interest. Now, this is a schematic in the rodent brain, so we work with rodent models. There are 5 dopamine receptors, as I mentioned, and the pathway highlighted here, sorry, I don't have a pointer, is the mesolimbic mesocortical dopamine neuronal system, which is the major mediator of many of the functions that I mentioned, and also are sort of the target that is thought to be aberrantly functioning in these neuropsychiatric disorders. And of particular interest is a region called the nucleus accumbens, which is part of the striatum. Now, dopamine D1 and D2 receptors are the most abundant of the dopamine receptors in brain. And it used to be thought, decades ago that one neurotransmitter, one receptor, one function, this very hugely simplistic model of neuronal function simply isn't true when one has a chance with more appropriate tools to take a look at what's happening in brain. Here, for instance, these are validated antibodies. So the tools have to be validated. Antibodies for the D1 and the D2 receptors, which are the most abundant in brain. And shown here in the top panel is prefrontal cortex region, showing that these receptors do co-localize in some neurons. And then where the green and the red co-localize. And then you can see other neurons where there's just red. So clearly there are neurons that express only one subtype and others where the subtypes are co-expressed. And the bottom panel shows cultured striatal neurons. So again, in culture, I'm sort of honing in on a particular neuron that clearly expresses both receptors. And then you can see another neuron on the side of it that expresses only one of the two. So dopamine receptors fall into sort of 2 subcategories. There is the D1 type family that activates adenyl cyclase and the D2 family that inhibits adenyl cyclase. So one question that we wanted to answer when we discovered this, more than a couple of decades ago, if the 2 receptors are expressed in the same neuron, would they simply sort of cancel each other out, or is there some other purpose? So to cut a long story short, if you look at the middle panel there, the middle aspect of the diagram, you can see we discovered that the two receptors interact with each other to form a novel signaling unit that had a completely different signaling pathway, coupling to a different G protein and now mediating a different second messenger signal, namely calcium, whereas the D1 alone or the D2 alone work through modulation of cyclic AMP through adenyl cyclase activity. So we had to figure out what this new complex and the new signal were doing in brain, and mapping studies through the brain, again, rodent brain, and we've used non-human primate brain and also human brain to show that there's a particular focus of concentration within the nucleus accumbens. So you can see the blue region in the rodent model, and then the bottom shows the MRI of in human brain, showing the nucleus accumbens, which sits at the base of the striatal complex. Now, the nucleus accumbens is sort of the hub, the reward hub in the brain. So we were very excited to find this particular concentration of these complexes in the reward hub, thinking that we would show that it was the mediator of reward. But when we were able to activate it, again, having to sort of develop tools to selectively activate this complex without activating the D1 alone or the D2 alone components, we found to our amazement that it mediated aversion. So dopamine, if you remember, has been classically associated with reward in brain. So here we had found a receptor GPCR receptor complex that mediates aversion. And indeed, activating it induced anxiety and a depressive phenotype in animals, in rodents that we have worked with and with a collaborator in non-human primate. So activating it induces anxiety and depression. And now, again, talking about the appropriate tools, how do we block the activity of this complex, this heteromeric complex? Because there were no ligands that we could discover by screening a large number of dopaminergic compounds, we had to devise a method to disrupt the complex by using a peptide strategy after identifying the amino acids that interacted with each other to form the complex. And using this peptide, which appeared to have no other function in brain, we were able to relieve that anxiety and depression and find that this had rapid anxiolytic and antidepressant-like effects in various models commonly used in animals to induce this phenotype, such as chronic unpredictable stress and so on. Therefore, this complex we found mediates aversive, anxiogenic, and depressive-like behavior, and disrupting it induced the converse. Now, we look for sex differences. Now, you've heard in one or two of the other talks that the incidence of depression in females is 2 to 3-fold higher than in males, in humans. And in rats, it's very interesting. Just showing you one of the many tests that we have done where we put the rat in a forced swim paradigm, where they're forced to swim and the rats give up, and this sort of mimics depressive-like behavior. And we find that male and female animals behave quite differently in this. If you look at the second panel over, you can see right at the baseline that the females had a highly depressive phenotype already before they sort of figured out what was happening in this paradigm. So they had a depressive phenotype. They gave up very quickly. And we found that disrupting that heteromer with the peptide injected into the brain, into the CSF, would relieve that depressive behavior. And the bottom panel shows, I don't know how well it shows up, there are red dots that show the signal that detects just that heteromer, showing that in the female nucleus accumbens, it was much higher than in the male. So we're seeing that the female phenotype uh, in rodents mimics what we know in humans, namely that the depressive phenotype is sort of inherent as a basal state in the females, disrupting the heteromer, abolishes it, and accompanying it, we have a greater proportion of the neurons in nucleus accumbens expressing this D1, D2 heteromer compared to males. And indeed, this accumulation or the concentration of this heteromer in the nucleus accumbens, we were able to show in non-human primate as well as in human nucleus accumbens. Also, the response to chronic stress was, more dramatic in the females. And again, we found, I don't have time to go through all the data related to the heteromer in the female nucleus accumbens compared to the male. And we wanted to look at the responses to drugs of abuse. And we found that in adult brain, adult rodents, as well as in adult monkeys, when they are given a drug of abuse, such as THC, which is the active principle in cannabis, or drugs like cocaine, we get an upregulation of this complex. So this is a physiological mechanism by which the nucleus accumbens is trying to place a ceiling on the reward perception. So if you give a high concentration of any drug of abuse, you get an elevation of this heteromer, which attempts to place a brake on the reward that is perceived. Now, in direct contrast, in adolescent brain, we find that the concentration of heteromer is much lower. So this is a maturational process in which the adolescent rodent does not achieve the number of heteromer, the density of heteromer expressing neurons until they are adults. And we find that their perception of reward is much greater because this aversive, negative, brake-like effect of this heteromer is not present. So we think this, sort of exposes or lays clear that one of the mechanisms by which adolescents have a higher vulnerability to drugs of abuse may be that they may perceive reward in the same manner, but they don't have this negative influence that limits reward, and therefore they are more prone to developing addictions when exposed to such drugs in this sort of neurodevelopmental stage when the dopaminergic system is still developing to full adult maturity. Now, one of the effects that we looked at in adolescent brain is what happens when exposed to these drugs of abuse. And what you're seeing in green are microglia. So we can see that in nucleus accumbens, in addition to not having adequate amounts of this heteromer, which is aversive, we find that there's microglial activation by THC in the nucleus accumbens. And this is not seen in any other region of brain. We've looked in prefrontal cortex and hippocampus and in amygdala, for instance. So, and this activation of microglia obviously, causes the release of cytokines and reactive oxygen species, which probably sort of accelerate damage that occurs by drugs such as THC and causes sort of excessive synaptic pruning and degeneration. And so this microglia-mediated oxidative stress is part of what leads to the neurotoxicity. So you can see that adolescent animals or perhaps adolescent humans don't have this negative influence of the aversive component of dopamine signaling, will take more drugs of abuse and be subject to greater toxicity from this oxidative stress that can occur, causing synaptic degeneration. And we're in the process of looking at various neuroimmune regulators which control synaptic pruning. There are eat me signals and don't eat me signals that can cause preservation of phagocytosis, and we're in the process of working that out. Another interesting hypothesis that we have is when any drug of abuse is used, we get a release of dopamine in various brain regions. And dopamine itself also causes activation of neuroimmune and neuroinflammatory mechanisms. In particular, in amygdala, we see that astrocytes are activated. And what you're seeing, the signal in red on the right panel is GFAP, so glial fibrillary acid protein, which is a marker of astrocyte activation. I'm not going to say too much about it because Yalan Sun, who is one of the awardees of MITO2i, will talk more about it during her talk. So this is another mechanism by which only in adolescent amygdala and not in adult amygdala, and we've shown this in both rodent and monkey, that this astrocyte activation occurs, which is another neuroinflammatory mechanism that seems to sort of potentiate the process. So the reactive astrocytes in adolescent amygdala sort of propagate the damage, which is induced by THC, and another mechanism by which this continued drug addiction process or the consequences, the negative consequences of those drugs will be more manifest in adolescent brain rather than adult. So that's sort of a quick summary of some of the things of interest to us at the moment. And I'm listing here current and past lab members who have participated in this and our collaborator from Harvard. Thank you.